Hello, everyone, and welcome to today's webinar, Test and Differentiate COVID-19, Flu A/B, and RSV with one assay. My name is Gabriel Orama of Thermo Fisher Scientific, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Thermo Fisher Scientific. To learn more about their COVID-19, influenza, and RSV testing solutions, please visit thermofisher.com/COVIDFluRSV. This presentation includes 4 questions that will appear on screen. 2 will appear now, and 2 will appear at the end of the presentation, before the Q&A session. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many as we can at the end of the presentation. You may also submit any technical issues here if you have trouble seeing or hearing this presentation. This webinar is educational and thus offers free continuing education credits. Please click on the continuing education window at the bottom of your screen to obtain your credits. Now I'd like to welcome our speakers, Jelena Feenstra, Senior Manager of Global Communications, and Peter Friebe, Senior Manager of Clinical Applications. Jelena and Peter work at Thermo Fisher Scientific within Genetic Testing Solutions, the unit responsible for bringing one of the first COVID-19 assays to market as part of the response to the pandemic. This presentation will begin with an overview of the respiratory virus ecosystem and will then switch to the workflow of our Thermo Fisher's COVID-19 Flu A/B RSV combo kit. You can find a shortened version of the workflow and other collaterals related to the product in the application note hosted in the studio. Feel free to download at Take Hope Study. With that out of the way, let's pass the microphone to Jelena Feenstra. Jelena, you may now begin with your presentation.
Hello, thank you so much for the kind introduction. Today I will talk about why it is important to detect and discriminate the influenza A and B, SARS-CoV-2, and respiratory syncytial virus during flu season. Seasonal flu is a contagious respiratory illness caused by influenza viruses that circulate in all parts of the world. In the Northern Hemisphere, the flu season typically occurs in the colder months of the year, and the peak in the number of infections usually happens between December and February. Depending on the year, for example, in the US, between 3 and 11% of the entire population suffers from a symptomatic flu infection, but a large proportion of infected individuals are actually asymptomatic. And here are some statistics regarding the number of cases, hospitalizations, and deaths resulting from flu each year in the US. Seasonal flu leads to 140 to 810,000 hospitalizations and 12 to 61,000 deaths per year in the US. And these numbers are fairly similar in the EU or the European Economic Area, with estimated 4 to 50 million cases and up to 70,000 deaths per year. Coronavirus disease 2019, or COVID-19, is an infectious respiratory disease caused by a novel coronavirus, SARS-CoV-2, that emerged at the end of 2019 in Wuhan, China. And on March 11th last year, the WHO had declared the SARS-CoV-2 outbreak as a global pandemic. By today, more than 228 million cases have been confirmed, with over 4.5 million people who died of COVID-19. And this graph on the right represents the number of confirmed cases per week in each part of the world from the beginning of the pandemic to mid-September 2021. And on the left, you can also see the total number of cases per each region. Now, let's look at the causative agents of these two diseases. So the seasonal flu is caused by influenza A and B viruses, and these viruses belong to the Orthomyxoviridae family of RNA viruses. And there are, in fact, 4 types of influenza viruses, A, B, C, and D, but it is, in fact, the A and B which are responsible for the seasonal flu infections. The influenza C infections generally cause mild illness and are not thought to be involved in the seasonal flu epidemics. And influenza D viruses actually infect cattle and are not thought to cause illness in people. So the genome of influenza viruses is RNA, and it consists of 8 negative single strands, so it's a segmented genome. And the structure of the viral particle is actually represented in the image here, and you can see 2 proteins on the surface of the influenza viruses, which are particularly important, and these are the hemagglutinin, which is shown here in blue, and neuraminidase, which is shown in red. And the hemagglutinin binds to the sialic acid, which serves as a receptor on the surface of respiratory epithelial cells and enables the virus to enter the cells. And the neuraminidase, on the other hand, facilitates the release of the viral particles from the infected cells and their spread. On the other hand, if we look at, the COVID-19, it is caused by SARS-CoV-2. And while SARS-CoV-2 is also an RNA virus, like the influenza viruses, it belongs to a different family. So these are coronaviridae. The genome of the SARS-CoV-2 virus is not segmented, so it is a single positive strand of RNA, and there is only one strain of the virus with different viral variants. And if we look at the structure of the viral particle, we can see that the surface of the virus is covered mainly in the so-called S, or the spike protein, which binds to the ACE2 receptor on human cells and enables the virus to infect them. So, as I mentioned in the previous slide, the SARS-CoV-2 virus is a single strain, while influenza A and B have various subtypes. So in case of influenza A, the subtypes are defined by the 2 surface proteins that I already mentioned, the neuraminidase and the hemagglutinin. And there are 11 types of neuraminidase and 18 types of hemagglutinin, which potentially gives 198 types of influenza A viruses. However, only 131 types have been detected in nature so far. And the currently circulating subtypes of influenza A are the H1N1 and the H3N2. The H1N1 is related to the 2009 swine flu pandemic. The influenza B virus has 2 lineages, the Yamagata and Victoria, and normally displays a slower change of genetic and antigenic features compared to influenza A. There was a misconception that the flu B viruses cause a milder disease, but since then several studies have shown that the influenza B can cause equally severe disease as influenza A. And currently, the seasonal influenza is a mix of influenza A, H1N1, A, H3N2, and the 2B lineages. In this graph on the right, you can see the surveillance data from the previous 5 years, and you can see that the percentage of each of the contributing influenza subtypes varies per year, and also that, in some years, some subtypes will appear earlier or later than the others. So let's look at how seasonal flu and COVID-19 are transmitted and how does the incubation time and clinical presentation compare. So both flu and COVID-19 are easily transmitted through direct contact and droplets, but also indirect spread and aerosols play a role in spreading the viral particles. COVID-19 is considered to be more contagious, and also more super spreaders have been observed with COVID-19. The person infected with the virus is usually contagious 1 day before the onset of symptoms and up to 1 week after the symptoms start in case of flu. But in case of COVID-19, one can be infectious 2 days before the symptoms appear and at least 10 days after. So in fact, people are contagious over a longer period of time in case of COVID-19. The symptom onset usually occurs in case of flu 1 to 4 days upon infection, so quicker, while for COVID, the incubation can take up to 14 days. And one thing that both viruses have in common is that infected people can be asymptomatic or have mild, moderate, or severe disease. Now, once a patient gets symptoms of a respiratory illness, How do we actually know if it is COVID or flu? And the fact is that COVID-19 and influenza cannot be differentiated based on symptoms only. If we look at the clinical presentation, usually a patient will have chills, fever, sore throat, headache, runny nose, general, tiredness. And in children, very common are vomiting and diarrhea. But these symptoms are common for both diseases. Now, of course, there are also some differences. So, for example, a loss or alteration of smell and taste has been associated with COVID-19. And also patients suffering from COVID have been shown to have sometimes cytokine storm, or in certain cases, some blood clots. And in children, the multisystem inflammatory syndrome has been described. On the other hand, patients suffering from flu more often get a secondary bacterial infection, and also younger children are at a higher risk of complications in case of flu than COVID. So actually, in case of both COVID and flu, a certain proportion of patients will develop a severe disease, which is then characterized by pneumonia, acute respiratory distress syndrome, and so on. And in particular, people with chronic conditions such as diabetes or cardiovascular disease may suffer from complications, as they are the risk group together with the people 65 years and older. But the fact of the matter is that if one wants to differentiate whether a patient presenting with these symptoms has flu or influenza, testing needs to be performed in order to confirm the diagnosis. So the CDC has actually developed recommendations on testing strategy when SARS-CoV-2 and influenza are co-circulating. So basically, if a patient comes in with symptoms of an acute respiratory illness and the patient requires hospitalization or is a resident of a nursing home, for example, it is recommended that the multiplex nucleic acid detection assay for influenza A, B, and SARS-CoV-2 is performed if available. Of course, the viruses can also be tested individually, but the multiplex test can offer faster results. And if the patients do not require hospitalization, it is recommended that they get tested by PCR for SARS-CoV-2, and that the test for influenza is also performed either as a single test or in a multiplex assay when available, if the results will influence the clinical management or steps taken to control the spread of the infection. Now, what is very important to remember, and is particularly highlighted in the CDC testing recommendations, is that the co-infections of influenza and SARS-CoV-2 can occur and do occur, and that if we do a single test for COVID first, for example, and it is positive, that does not automatically exclude influenza infection in the same individual. And this is the exact reason why the multiplex testing is recommended, and especially in hospitalized patients or nursing home residents, in order to prevent cross-infection in these settings, as patients having COVID only must, of course, be separated from patients that are suffering from a co-infection with flu. So, in cases of mild illness of both COVID and flu, the treatment is mainly focused on symptom management and includes bed rest, isolation, avoiding contact with other people. In cases with severe disease or in people at high risk of complications, supportive medical care is given to manage symptoms and prevent complications or manage them. However, treatment strategies in all other cases are different for COVID and flu. And of course, the treatment algorithms may vary a little bit by country and local recommendations, but this is the sort of general overview. So outpatients at high risk of disease progression with COVID-19 can receive anti-SARS-CoV-2 monoclonal antibodies. And in hospitalized cases on supplemental oxygen, remdesivir, dexamethasone, or both agents are given to patients. In patients who require oxygen delivery through a high-flow device or ventilation, In addition to dexamethasone, baricitinib or tocilizumab are applied. And treatment decision for COVID-19 actually does not depend on the viral variant with which the patient is infected. In cases with flu, antiviral drugs have been shown to have the best clinical benefit if given in the first 48 hours of the symptom onset, which is an additional reason why testing that can enable a quick and precise result is essential. There are several approved drugs for flu, such as neuraminidase inhibitors, which are oseltamivir and zanamivir, but also polymerase acidic endonuclease inhibitor baloxavir. In addition, antibiotics are prescribed for secondary bacterial infections, which we already mentioned are more common in cases of flu. The virus type or subtypes actually do not influence the treatment decisions. So even though clinically based on symptoms only, the two diseases are very similar, the clinical management of patients is very different, and correct diagnosis is very important in order to achieve better outcomes. Now, one thing that both influenza viruses and SARS-CoV-2 have in common is that they mutate. And in the case of influenza virus, there are two types of changes that are happening. The so-called antigenic drift, which represents minor changes to the genome, and then antigenic shift, which usually happens when one segment of the genome is replaced by a segment from a different influenza virus. And this usually happens in animals and is the way how strains that can give rise to flu pandemics arise. So as only influenza A viruses have animal reservoirs, they are the only ones that can actually give rise to a pandemic. There were 3 pandemics of flu in the 20th century, with the latest one in 2009, in which case the genes from North American swine, Eurasian swine, human, and bird flu were combined in the H1N1 PDM09 virus. On the other hand, SARS-CoV-2 virus accumulates mutations in its genome, most of which are actually affecting the S protein, which is the surface protein. And the emerging variants that contain a particular set of mutations are classified based on the risk they pose for the public health as variants of interest, variants of concern, and variants of high consequence. At the moment, there are 5 variants of concern circulating, which are referred to as alpha, beta, gamma, and delta, plus the alpha variant with the E484K mutation. And some of the variants share these key mutations relevant to the function of the virus. How do we prevent the spread of COVID and flu? So in both cases, personal protective measures are recommended, including hand washing, avoiding to touch your face, nose, or mouth, or contact with infected individuals. And during the COVID-19 pandemic, countries have implemented additional strict public health measures prior to the availability of the vaccine, including social distancing, travel bans, etc. Now, of course, vaccinations play a major role in preventing disease, and for flu, annual vaccination is recommended for risk groups. As we have seen, the viruses mutate and different subtypes may be present each year. So the flu vaccines are annually updated based on the surveillance data on influenza viruses from the previous flu season. And normally, because this is sometimes not the best predictor of which subtypes will be circulating next year, the flu vaccine efficacy is reducing the risk of flu illness varies between 40 and 60%. And several vaccines are available in the EU. The most common are trivalent or quadrivalent injected inactivated influenza vaccines. The COVID-19 vaccines have been developed in really record time in 2020, as you already know, and currently there are 4 different vaccines that have been authorized for use in Europe by the European Medicines Agency. And the current recommendation is to vaccinate everyone 18 years and older, with 2 vaccines also authorized for children 12 years and older. And the vaccines are based either on RNA technology or viral vectors. And although SARS-CoV-2 mutates, it is still unclear whether updates to the vaccines will be needed due to novel emerging viral variants. Several currently circulating variants of concern are associated with slightly reduced efficacy of the currently available vaccines, but so far it is considered that the vaccine efficacy is high. Now, besides COVID and flu, there are, of course, other respiratory viruses which can cause similar symptoms. And today I would just like to mention the respiratory syncytial virus of the RSV, as this is one of the leading causes of respiratory disease globally and also shows seasonal activity similarly to flu in autumn and winter months. And actually, the symptoms are usually cough, runny nose, fever, loss of appetite, and also severe disease may develop, causing pneumonia or bronchiolitis. So as you can see, these symptoms are actually very, very similar or overlapping with flu and COVID-19. The risk groups for severe disease are actually young infants and older adults, or people with chronic diseases or weak immune system. RSV is estimated to lead to around 60,000 hospitalizations in kids younger than 5 and more than 170,000 hospitalizations in adults over 65 years of age annually, with 14,000 deaths among the adults 65 years and older, for example, in the United States. And the treatment is focused mainly on symptom management, while vaccines for RSV are in development. So you can immediately see that there are a lot of similarities in clinical presentation of RSV to COVID-19 and flu. And on this slide, I would like to show you some of the surveillance data from the EU, where you can see, for example, on the upper panel, how the RSV or the seasonal activity of RSV in the past couple of years, with peak of infections occurring usually in the last or the first week of the year during the winter. And on the graph below, you can see seasonal flu, which is active in the same period of the year, what we refer to as the flu season. But the peak of infection varies from December to March. And for example, in the last 12 years, which are shown on this slide, or on this graph, the peak was most often seen in February, so in weeks 5 to 7. But what I want to bring your attention to is this last year, which is highlighted in pink, where you can see that the data initially showed that actually there was no flu or RSV circulating during the usual flu season. However, then cases started to peak at week 11 for flu and week 13 for RSV, which is much later than usual. And this timing is very relevant with regard to the public health measures that were in place related to the COVID-19 pandemic that I will talk about in the next slide. As the COVID-19 vaccination is progressing worldwide, we can see that some countries like the United Arab Emirates, Spain, or Portugal have managed already to fully vaccinate 80% or more of their population. But worldwide, only 42% of people have received a single dose, and actually only 30% have been fully vaccinated. And of course, the rate of vaccination has had an impact on the public health measure stringency shown in these graphs. So if you look here, the dark red color corresponds to high stringency, meaning social distancing, travel bans, mask wearing, etc. And you can see that many countries still had strict measures in place, for example, in February this year. But then as the vaccination for COVID-19 progressed, most of the countries were starting to ease the restrictions in February or March. And this exactly corresponds to the sudden peak in cases of flu and RSV that I have shown you a couple of slides earlier. So it is actually clear that all the public health measures that were put in place for COVID-19 were in fact very efficient in preventing the spread of other respiratory viruses like flu or RSV. But as the restrictions are easing, it is expected that the surge in cases of flu and RSV might occur, as already seen in some parts of the world. Now, in fact, the Academy of Medical Sciences in the United Kingdom and also the WHO have published reports looking ahead to winter 2021-22 and beyond, where they state that higher activity of influenza and RSV viruses is anticipated, and that easing of social distancing and other measures may lead to surge in infections. And in addition, we have to consider that there is a decreased population immunity due to non-pharmaceutical intervention and lower exposure to influenza and RSV in the previous season, which might also play a role. Of course, there is also limited data available for influenza strains in the previous season for vaccine design, as we saw that there were not so many flu cases, which of course increases the likelihood of influenza vaccine mismatch for the upcoming season. And finally, I would like to conclude my talk with the two statements on testing recommendations coming from the European Centre for Disease Control and the Academy of Medical Sciences in the UK. So all patients with acute respiratory symptoms in hospitals and other healthcare settings and all specimens from sentinel primary case surveillance should be tested for both SARS-CoV-2 and influenza during the influenza season to monitor incidence and trends over time. And then, given overlapping symptoms, routine multi-pathogen testing for SARS-CoV-2, influenza, and possibly other respiratory infections is important for surveillance, treatment decisions such as timely use of antivirals for influenza, minimizing isolation times, and avoiding and reducing rates of transmission. We strongly support multiplex testing. So with this, I would like to thank you for your attention, and I'm happy to hand over to Peter Friebe, who will present the testing solutions Thermo Fisher Scientific has developed to enable the laboratory professionals to meet the challenges of the upcoming flu season. Thank you.
Thank you, Jelena, for this excellent and very informative overview on the importance of detecting and differentiating flu A, B, RSV, and SARS coronavirus 2 during the next flu season. In the second half of this webinar, I want to introduce you to the TACPAS COVID-19 Flu A, B, RSV Combo Kit, which is Thermo Fisher Scientific's solution to meet the demand for respiratory testing during the next flu season. My name is Peter Friebe, and I'm a Senior Manager for Clinical Application and Marketing Support. The second half of this webinar will be clustered into a brief introduction to Thermo Fisher Scientific, the company. I will then highlight the TACPAS COVID-19 Flu A, B, RSV Combo Kit and will focus on the workflow, the turnaround time, and the performance of the kit. And at the very end, I will give you a brief introduction into our new AB Platinum Instrument Service Plan. Thermo Fisher Scientific is the global leader in COVID-19 testing solutions. Over 50% of all COVID-19 testing performed is using Thermo Fisher technology. We were able to upscale COVID-19 test manufacturing to over 20 million tests per week, and if need be, we can even increase this capacity. We also offer a secured supply chain to reliably meet global testing demands of all the consumables, test kits, instruments needed for our clients to perform all the tests needed. We can do so because Thermo Fisher Scientific is vertically integrated, and all the companies that are listed on top of this slide are part of Thermo Fisher Scientific. We also offer a 24/7 service and support on all our consumables, instruments, and the software. And with that, I want to introduce you to the TACPAS COVID-19 Flu A, B, RSV Combo Kit, which is CE-IVD marked and for in vitro diagnostic use. The TACPAS COVID-19 Flu A, B, RSV Combo Kit is an all-in-one CE-IVD marked RT-PCR respiratory test designed to increase lab efficiency. It can, in a single test, detect and differentiate between SARS-CoV-2, Influenza A, B, and RSV, and it can also detect co-infections. It is affordable and scalable. The workflow allows to increase testing throughput, therefore increasing lab efficiency. It is a highly sensitive and specific detection kit with a very robust performance to detect SARS coronavirus 2, flu A/B, and RSV. And our TAGPass COVID-19 flu A/B RSV combo kit also comes with our pathogen interpretive software that will automatically validate each test run and create a report for each of the specimens tested. The workflow of the TAGPass COVID-19 flu A/B RSV combo kit is shown on this slide. I will go over each of the steps shown on this slide in more detail in my next slides, but want to start giving you a general overview. Our lab will first receive the samples. The samples then need to be prepped. The real-time PCR is prepared and run, and then the interpretive software will calculate the results and create a report. Sample types that are supported by the kit are nasopharyngeal swabs. Once the samples are received by the lab, they need to be prepared. We do recommend to use our MacMax Viral Pathogen 2 Nucleic Acid Isolation Kit in combination with our automated Kingfisher Flex purification system for streamlined sample preparation. Please note that before preparation of the sample, the MS2 bacteriophage is added to each specimen. MS2 bacteriophage will act as an extraction and process control for each sample. Once samples are prepared, the real-time PCR is set up on a 96-cell plate using the TAGPass COVID-19 flu A/B RSV combo kit. The PCR is run on the 7500 fast real-time PCR system. Once the run is finished, the interpretive software, the pathogen interpretive software, will automatically interpret test result. It also performs a quality control on all the controls on the plate, which is at least one positive control, one negative control, and the MS2 bacteriophage process control in each sample. The pathogen interpretive software will also generate an interpretive report for each of the specimens included in the test. The software also allows to export the results in a file that is compatible with your LIMS or LIS system available in your laboratory. The pathogen interpretive software can be installed on any qualified computer. It can be co-installed on a computer that already runs our COVID-19 interpretive software. And the pathogen interpretive software supports analysis of datasets from multiple qPCR instruments that can be sent seamlessly over network to the computer that has the pathogen interpretive software installed. The user interface of the pathogen interpretive software is very user-friendly. It is shown here on this slide. To the right upper corner, it will display the 96-cell plate and the samples that are included on the test run. The samples are either marked with a plus or minus, depending whether a virus was detected or not, and hovering over a well will show the amplification plots to the left of the respective targets. On the bottom part of the user interface, you can also see a table with all the samples tested and the results for each of the samples. Again, the results can be exported in a LIMS compatible file, or you can export it in our interpretive report. The interpretive report will include information about the run itself. It includes information about the date of the run or the time of the run and the instrument used to perform the run. It includes information about the positive and negative control, and then it includes the results for each of the specimen tested, and it will highlight whether or not a virus was detected in each specimen. Now that I've introduced you to the workflow, I also want to give you an overview of the turnaround time to perform the assay. The overall turnaround time from sample to result is about 3 hours. It takes roughly 45 minutes to receive 94 samples that can be tested on a 96-well plate. Please keep in mind that one well is dedicated to a positive and one well is dedicated to a negative control. Sample prep, if carried out with our Kingfisher Flex system, takes only 25 minutes for a 96-well plate. Preparing the PCR takes about 30 minutes, and running the PCR takes 82 minutes. and the report is ready within 5 minutes using our pathogen interpretive software. To perform the PCR from setup, running the PCR and getting the result only takes about 2 hours. As Jelena already mentioned, the next flu season might be a more severe one. It might become necessary for laboratory to increase throughput of testing. The TAGPath COVID-19 Flu A/B RSV combo kit is designed to allow maximizing throughput by staggering batches of samples as it's indicated on this slide. The example on this slide uses a laboratory that is equipped with one Kingfisher Flex, 2 RT-PCR system, and it uses 2 full-time employees per 8-hour shift, and that allows to run up to 2,700 tests per 24-hour day. You can see in dark blue the staggering of a batch of samples that starts with receiving the samples by the first employee. Once the samples are received, they are transferred into the Kingfisher Flex for automated sample purification. Once purified, the second employee prepares the PCR and starts the PCR run. At that point, the first employee is already receiving the second batch of samples and transferring the second batch of samples into the Kingfisher Flex. Once the second batch of samples is purified by the Kingfisher Flex, the second employee will start preparing the PCR and run the PCR. This kind of staggering allows to maximize the throughput, and as mentioned earlier, it allows up to 2,700 tests per 24-hour day. I now want to introduce you to the performance of the TAGPath COVID-19 Flu A/B RSV combo kit. I want to start with the limit of detection, which is expressed as either tissue culture infectious dose 50 per ml, or, to simplify, as genome copy equivalence per ml. For SARS coronavirus 2, the limit of detection is 50 genome copy equivalence per ml, or short GCE per ml. For Influenza A/B, the limit of detection varies between 350 and 1,250 GCE per ml, and that depends on which strain is tested. For RSV A/B, the limit of detection is 200 GCE per ml. We also performed wet lab testing with different strains for Influenza A and Influenza B. For Influenza A, we tested 10 different isolates, focusing on the predicted dominant strain for the next flu season, which is the H1N1 and the H3N2. For Influenza B, we tested 5 isolates, including the Yamagata and the Victoria line. We tested all the samples at a concentration that is very close to the limit of detection, and all isolates were tested in triplicates. The TAGPath COVID-19 Flu A/B RSV combo kit detected the virus in all samples tested. We also performed wet lab testing for RSV. We tested 3 subtypes. We tested 3 isolates for subtype A and 3 isolates for subtype B. Again, the tests were carried out with a concentration that is very close to the limit of detection, and the TAGPath COVID-19 Flu A/B RSV combo kit detected the virus in all samples tested. We also performed cross-reactivity tests to ensure that the TAGPath COVID-19 Flu A/B RSV combo kit is highly specific to SARS coronavirus 2, Flu A/B, and RSV. We tested all the infectious pathogens shown on this slide, and we did not detect any cross-reactivity. The TAGPath COVID-19 Flu A/B RSV combo kit is a multiplex assay, meaning it can detect RSV, flu, and SARS coronavirus 2 in the same sample. It can also detect co-infection. And to ensure that there is no competitive interference, we performed a study in which we tested samples that were spiked with 2 viruses, one at a very high titer and one at a very low titer. To the left, you see the results of samples we tested that were spiked with a very high flu titer and that had a very low titer of SARS coronavirus 2, a titer that is close to the limit of detection. As you can see by the amplification plots shown on this slide, the assay detected the flu target in all samples, which is shown with the red amplification curve on the left amplification plot. It also detected SARS coronavirus 2, shown here with the green amplification plot and labeled COVID low. Additionally, it detected the MS2 bacteriophage process control. RSV was not detected in the sample because it was not present in the sample. On the right-hand side, you see the amplification plot for samples that were spiked with a high titer of RSV and that had a very low titer of influenza close to the limit of detection. And again, the assay detected influenza at the low titer, even in the presence of a high titer RSV co-infectant. We also performed a clinical study using retrospective samples. The clinical study was carried out with the TACPAS COVID-19 Flu A/B RSV combo kit and a comparator for either SARS coronavirus 2, Influenza A, B, or RSV A/B. Shown on this slide are the positive percent agreement and the negative percent agreement. Just as a reminder, the positive percent agreement reflects the percentage of samples that tested positive with the TACPAS COVID-19 Flu A/B RSV combo kit and that also tested positive with the comparator, whereas the negative percent agreement reflects the percentage of samples that tested negative with the TACPAS COVID-19 Flu A/B RSV combo kit and that also tested negative with the comparator for the respective target. The positive percent agreement for SARS coronavirus 2, Influenza A/B, RSV A/B is over 98%. In case of Influenza A/B, it is 100%. The negative percent agreement for RSV A/B is about 93%. For Influenza A/B, it's between 96 and 97%, and for SARS coronavirus 2, it's 100%. Although a CE-IVD marked in vitro diagnostic device does not have a guideline what percentage numbers needs to be hit, the US FDA actually provides that kind of a guideline, and the percentages shown on this slide exceed the requirements for the US FDA and overall demonstrate the excellent clinical performance of the TACPAS COVID-19 Flu A/B RSV combo kit. With that, I want to summarize. The TACPAS COVID-19 Flu A/B RSV combo kit targets SARS coronavirus 2, Influenza A/B, and RSV A/B. It can detect and differentiate between all those three viruses in one sample. It can also detect co-infections. It uses the MS2 bacteriophage as an internal process control. A positive control is included in the kit. It uses nasopharyngeal swabs as a proof sample type. We do recommend the MagMax viral pathogen nucleic acid isolation kit in combination with our automated Kingfisher Flex purification system for sample preparation. The approved real-time PCR system is our 7500 fast real-time PCR instrument. The turnaround time from sample to result is about 3 hours, and from extracted RNA to result is just under 2 hours. The combo kit comes with our pathogen interpretive software that will automatically validate each run and prepare a report for each of the specimens included in the test. The TACPAS COVID-19 Flu A/B RSV combo kit is CE-IVD marked and for in vitro diagnostic use. If you are interested in our TACPAS COVID-19 Flu A/B RSV combo kit, you can go through thermofisher.com/COVID-19-flu-RSV to find more about the kit itself and to also find the ordering number, which is A49867. We currently offer the TACPAS COVID-19 Flu A/B RSV combo kit in a 1000 reaction format. And the kit includes all primer and probes for the detection of the SARS coronavirus 2 Flu A/B RSV and the internal process control MS2 bacteriophage. The process control is also included in the kit. It also includes the positive control and a positive control dilution buffer, and it does include the TACPAS one-step multiplex master mix which includes the enzyme for the reverse transcriptase PCR. Last but not least, I also want to introduce you to the AB Platinum Service Plan. The AB Platinum Service Plan is offered to our CE-IVD COVID-19 customers, and it covers qPCR and Kingfisher system. We do offer a 98% uptime guarantee. Of course, terms and conditions apply. The AB Platinum offering includes a qualification system. Each system is qualified post-service and repairs. It offers a priority technical support that is available 24/7 year-round, that includes weekends. It also offers a rapid on-site response, a guaranteed next business day on-site response. It includes a 2-hour remote field application specialist consultation. It also covers all plant maintenances as required. It includes a comprehensive repair coverage. It's an all-inclusive repair, and also a digital remote support. It covers our Kingfisher Flex purification system, and it covers our QuantStudio 7 in 384 well format. It covers our QuantStudio 5 in 96 well and 384 well format. It covers our QuantStudio 5 DX. It covers our 7500 Fast DX, and it covers our 7500 and 7500 Fast real-time PCR system. If you want to learn more about our AB Platinum support program, please visit thermofisher.com/uptime. Please also note that the AB Platinum is only available in certain regions and countries. With that, I want to thank you for attending this webinar, and I open the session for the Q&A. I see a question in the chat. How can I order the kit? As I mentioned, that is very easy. You can just go to thermofisher.com COVID flu RSV and request a quote, and we will follow up with you.
All right, thank you so much, Peter. Okay, it looks like our next question is, how common are co-infections of COVID and flu?
So let me take that one. Thank you. So yeah, that's actually a great question. And at the beginning of the pandemic, there were some reports. For example, there was a paper reporting on around 300 patients from Wuhan in China that were in the hospital early in the pandemic during January and February 2020 when the flu was still circulating. And they actually reported that around half of the patients with COVID had also influenza A, and around 5 to 10% of the patients also influenza B. But then, some of the reports that came later actually said that COVID and flu co-infections occur somewhere between 0.5% and 5% of the cases. And for example, despite low circulation of RSV, an article in JAMA reported that actually also SARS-CoV-2 and RSV co-infections also occur. And overall, it seems to be the consensus that, RSV and influenza A are the commonest viruses to occur together with COVID. Now, most of these studies were published when there was this really big drop in circulation of other respiratory pathogens due to all these measures that were taken to combat the pandemic. And, with that being said, the concerns really are about the upcoming flu season, with the easing of the social distancing and mask wearing and travel opening up. If the flu season this year looks different, how will the co-infections look like? And therefore, many of the agencies are actually recommending, the multiplex testing for several viruses.
All right, thank you so much, Jelena. Our next question, let's see. Can the COVID flu RSV kit substitute the COVID-only test? Would you like me to repeat the question?
Peter, would you like to take this one or shall I?
Oh, I know, I take it. I'm sorry, I had some internet connectivity issues. Yes, our TACPAS COVID-19 flu A/B RSV combo kit is intended to detect SARS coronavirus 2. It can also detect and differentiate between influenza A, B, and RSV in the same well, and that makes our kit a valuable multiplex test or kit for the upcoming flu season.
Okay, great. Thank you. Now, our next question here, how is the severity of a flu season defined?
So, yeah, thank you. Maybe I can take this one. Yeah, so, it really depends. But for example, the CDC has very clear criteria, and usually the severity of the flu season is classified as low, moderate, high, or very high. Normally, the agencies would use the data such as, for example, percentage of patient visits to outpatient clinics for an influenza-like illness, or the rates of influenza-associated hospitalization, then percentage of deaths resulting from influenza or pneumonia that occurred during each season. And, normally the flu season severity depends on the viral subtypes which are circulating each year, as I explained. So there is really a big question coming up ahead of the following season, what will be the subtypes and how severe will the flu season be, considering also this, reduced general immunity of the population.
All right, thank you so much. Our next question, how much sensitivity is the kit?
I will answer that. So there's an analytical sensitivity, which is equal to the limit of detection. And the limit of detection for SARS coronavirus 2 is 50 genome copy equivalents per milliliter. For RSV, it is 200 GC or genome copy equivalents per milliliter. And for flu, it is between 350 and 1,250, and that depends on which strain is tested. If the question is more related towards the clinical sensitivity, we express that as positive percent agreement, and it's over 98% for each of the targets. It's 100% for influenza A/B.
Thank you so much, Peter. Okay, next question. You mentioned a surge of RSV cases were observed in other parts of the world. Could you please elaborate on that?
Yeah, so as I said, there was really no activity of flu and RSV during the usual winter months last year. And then suddenly in summer this year, many countries have reported RSV outbreaks. So, for example, in the in the US, the CDC data showed that the levels of RSV are actually back to 2019 levels, but at a different time of the year. So normally we would see this peak in December, but now we saw a peak in August. And for example, Public Health England has also reported data that there was a surge of RSV infections during the 2021 summer. So that seems to be the trend in the Northern Hemisphere.
All right, thank you, Jelena. Okay, we've got time for one more question. Next question, I would like to ask, is this kit more preferable to research using or clinical diagnosis?
So the TACPAS COVID-19 flu A/B RSV combo kit is CE-IVD marked, and it is for in vitro diagnostic use, so it would be for medical application.
Thank you so much, Peter and Jelena, for your time today. Do either of you have any final comments before we close?
So yeah, I would just like to thank Peter for this great overview of the kit, and we are all concerned of what lies ahead of us against in the upcoming months. So yeah, I guess we are all working towards being prepared the best way we can and enabling the labs to combat the upcoming flu season in the COVID pandemic in the best way possible. So thank you for your attention.
All right. Well, thank you both again for your time today and your important research. We would also like to thank LabRoots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. As a reminder, questions we did not have time for today and those submitted during the on-demand period will be addressed by our speakers via the contact information you provided at the time of registration. This webcast can be viewed on demand, and LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time.